Hello, everyone. This is Dr. Jackson, and this is the lecture for Chapter 8, Hypothesis Testing. I just want to let you know that this particular lecture is going to come in several parts. As you can see here, there are quite a few learning objectives. It's a lot of stuff. However, it actually all goes into, I guess, one thing that you're doing, which is hypothesis testing. But hypothesis testing has quite a few parts to it. So that's why you see quite a few learning objectives here. But and of course that's the chapter outline for those of you that like to maybe kind of outline how you're going to take your notes. And this is essentially the order that I will go in with this particular lecture. And I think that this lecture will come in at least 3 parts. I'm thinking it's going to be 4, maybe more. Not sure. We'll see how long it takes me to get through each section. I think I've broken it up into, let me go back. I think I've broken it up into, in terms of the lecture itself, into at least 3 sections. The first section that we'll go over, we will, do an overview of hypothesis testing, what it is. Then I think at that point I stop and the 5-step model for hypothesis testing. I think that's its own video. Choosing a one-tailed or two-tailed test is a video. I think the student T's distribution may be in there as a video. That's part of where I talk about the one or two-tailed, and then the test of hypothesis for single sample proportions. So like I said, 3, maybe 4. If there are 4 sections, then there will be a 5th section. And that very last section is where I will go over and do the practice problems that I like to do. So like I said, 3 or 4 sections. I'm trying to keep these so that they're no more than maybe 30 minutes long. So that's why there will be so many sections, because there are quite a few slides here, but it's a lot of information. So on that note, what I would suggest you do is whatever it is that you use to take your notes, have all of that stuff ready to go, maybe even have some of your notes and information from previous chapters, because this is where a lot of the stuff that we've talked about before will start to come together. You'll see some of the stuff as it relates to, you've continued to see standard deviation ever since I introduced it. The normal curve, that's been in almost every single chapter. Some of the stuff that you did for confidence intervals, where you have to do the reverse to get the z-score and all of that type of stuff. A lot of that stuff that we've been learning up to this point, it's all about to come together. So you may need to go back and practice a few things, refresh your memory, so on and so forth. But that's definitely something you'll want to have your old notes as well as the book. If you have the book, have that ready to go. Okay. Oh, also, one of the things you may want to consider doing is if you've already read the entire chapter, great. If you haven't, and you are the type of person that watches my lectures and then you go back and read the book, whatever works for you. But one thing I would suggest is to go through all of this in sections. So go through the first part. If you've already read the book, great. Listen to the lecture for the first part in the first video, which is this video. Then go back and review the material in the book, and then maybe do some of that stuff as a practice in the lesson, and do practice problems on your own. Then do the same thing with the next section and the next section and the next section, okay? Because it's a lot, okay? It is a lot to try and remember and do all at one time. Okay, here we go. All right, so one of the things here, what we talk about first is significant differences. And you will hear me say statistical significance, or I'll say it was significant at the such and such level. Well, what we're talking about, or what I'm talking about when I say statistical significance, this is the determination that a relationship between 2 or more variables is caused by something other than chance, okay? So remember, one of the things that we've been talking about all this time is dealing with things whether or not something is, if the differences take place by chance, okay? So I think I will do yellow for now. So I'm gonna highlight. So the key thing that you'll wanna remember here is, okay, there we go. Is that, hang on one second, you guys. Okay, so yeah, the key thing that you'll wanna remember is differences don't, like, in order for something to be significant, the differences can't take place based on chance, okay? There we go. Also, statistical significance is something that we use to provide evidence concerning the possibility of something that we call a null hypothesis, which basically hypothesizes that there's nothing more than random chance at work in the data. And I'll talk more about null hypotheses or the null hypothesis in a moment, okay? But basically, when we're saying that the differences going on are not by chance, when we say that, so I'm going to highlight this so you can see what I'm talking about. When we say this, that differences do not occur by random chance, that's also called hypothesis testing. Okay, some people will say hypothesis testing is significance testing, but basically what we're trying to do is determine, it's a test of statistical significance. I was doing so well before I started to have trouble with that word. But you guys know I have trouble with it, so you'll hear me kind of stumble over it from time to time. But one of the things, something that's really important to think about when it comes to statistical significance, it's not something that we use just for social science research. So we don't just use this for research in criminal justice and criminology and all that stuff. It's also used in real world situations. For instance, it's often used for new pharmaceutical drug trials, to test vaccines, and in the study of pathology for effectiveness testing, and to inform like investors on how successful the company is at releasing new products. So it's used in the business world, it's used in the medical field, it's used in all types of places. So sometimes when you see these surveys and different things that are put out there and someone comes out and says, they start talking about these results, a lot of times in the media, they don't talk about things like statistical significance. Some of you may have heard about some of the medical trials that are going on right now. Some of the medicines and vaccines that are being put out there, and people talk about, well, it has a such and such percentage, or it works at the such and such percentage level, like 64% of the time, or in 64% of patients, it worked, blah, 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 blah. Well, one of the things that they have to also do is look at statistical significance, because we can say anything works, but does it work? And does the whatever it is that we're testing, is that outcome really happening, or is that outcome simply the result of random chance? Okay, so that is essentially what we mean, or what I mean when I say significant differences, or when I say statistical significance. Okay, which is ultimately what we're going to be doing here, testing hypotheses. We're testing hypotheses to determine whether or not whatever it is that we came up with is actually real. Are those differences real? Okay, and so I'm going to kind of demonstrate that with this example. It's on this slide and the next few slides. So you'll see it start to play out as I continue to go through this. But basically, here's the example. The education department at a university has been accused of grade inflation. So education, someone saying, hey, education department, you guys are inflating your grades. That's why education majors have higher GPAs than the general student body, okay? Ultimately, the GPAs of all education majors, what they say should happen is, the GPAs of all education majors should be compared with the GPAs of all students. But the thing is, there are thousands, not just 1,000, but thousands of education majors at this particular university. So that's far too many to interview. So maybe it's a really, really big university. And, just think about whatever state you're from, the big flagship university. For those of us here in Texas who are from Texas, you know that places like Texas A&M, UT, University of Texas, you know, Texas Tech, those are some of the bigger universities in the state. So imagine wherever you are, because I know not everyone is here in Texas, but wherever you are, just think about the big flagship university, the one that everyone knows. You know, I'm from the area that I'm from is where Bryan College, is where Texas A&M is located. So I'm going to use Texas A&M. So let's say that some folks, other departments at Texas A&M have said to the education department that they're accused of, you know, they're saying that you guys inflate your grades. You're giving your students grades that are too high. And the thing we know, Texas A&M is a really large school. So they have thousands of education majors. That's way too many education majors to interview. Plus, you also have to think about the, you know, majors from other departments. So not just education majors, maybe you have sociology majors, CJ majors, you have math majors, biology, you know, ag, all these different departments and people like with their majors or whatever. You've got to compare those GPAs. So we're talking about lots and lots of students. This is where, of course, sampling comes in, because, you know, how can this discrepancy be investigated without actually interviewing all education majors, or without going and getting the GPAs of every single major, every single student in every single major, and then doing all that comparison? That would take forever to do. Both things would take forever to do. So what happens is you get a sample. You know, this is the information this is the information for a sample taken from the population information that was on the previous slide. So the information in the box is the sample. So let me go write that out for you, or just at least say sample. Give me just a second, folks. Okay, so this right here, there we go. All right, so I haven't written on this thing in a while. I'm usually in the OneNote, so let's see if I can do this and do it in a straight line. So this is your sample. All right, this right here, the 270, that is actually going to be your, let me make sure. This is going to actually be your population mean. All right, so remember, okay, I'll just erase everything. Remember that for the population mean, this funny looking M, that is the symbol for the population. All right, remember, don't forget that. This little thing right here, that's the symbol for the population mean. All right, some people say it looks like a strange U. I say a funny looking M. All right, so that is your population mean. This information right here, that is for your, oh no, that is for your sample, okay? All right, so that is for your sample. I'm not even gonna write it, we'll just leave it be. So in the sample has a mean of 3. The the sample standard deviation is 0.70, and the number of people taken in the sample is, it looks like it's 117, okay? So this is the information that you're gonna need in terms of the population and the sample, all right? So again, not gonna go through and say all the information for the problem, you know, all the information that was on the previous slide, but, oh, I must have accidentally erased everything. Anyway, you guys get the gist. You know what the population mean, which is the population parameter. We know that that's 2.70. Okay. This right here is the sample mean, sample standard deviation. So sample mean, sample standard deviation, and that's the number. That's the number of people that were included in the sample. That information is going to be really key and important when it comes to testing the hypothesis. Notice that we have not been given the population standard deviation. All right, keep that in mind. Remember that. All right, so at first glance, looking at this, you would say, oh, okay, well, yeah, there is a difference, because remember, this is the population, this is the sample. So at first glance, we'd look at it and say, yeah, there's a difference. The folks who have, let's see, the sample of education majors, their mean is 3. And that's more than the population mean. which is 2.70. So yeah, of course they're different. However, what we have to keep in mind is that since this information was taken from a sample, we can't just say it's because of grade inflation. There is the possibility that the difference is due to random chance. And so statistical significance is what's going to tell us if that difference is real or not. Okay, this is how you test the hypothesis. Remember that the hypothesis is basically that education majors, well, the problem itself is education majors, their grades are inflated. So the education department is saying the education majors that their grade is inflated. So the hypothesis would be education majors have a higher GPA than all other groups. Okay, what we want to know, are those differences between the two groups real? So is this difference between your population mean, which is 2.70, and the sample mean, which is 3.00, we want to know, is that difference real? Okay, or is it just random chance that those differences came about? And a lot of the reason we do this is because, like it says here, because we're working with a random sample, okay, a random sample, and that basically means we don't have the information for all education majors. So we have to account for that. All right, so the two explanations for the difference could be that it is either, as number one indicates, that there's no real difference, or that the difference happens to be so small and trivial, and that it's really and truly due to chance, to random chance. Or it's number two, which is basically the difference is real and not due to chance. Again, this is statistical significance. So the question then becomes, how do we determine this? How do we determine whether it's number one or number two? And this is where hypothesis testing comes into play. And one of the things to keep in mind is we have these two explanations, but only one of them is valid. Only one of them is the actual case. And again, this is where hypothesis testing comes into play. We want to find out, is the difference, so like we see here, is this difference trivial and caused by random chance? Okay, so that's number one. We want to determine, is the difference trivial and caused by random chance, or is the difference real? Okay, is this difference real? In other words, is it significant? That really and truly education majors really are different from all other majors, from all other students. That's what we're trying to find out. So to do that, that's when we do the hypothesis testing. Now, before we get into hypothesis testing, I want to make sure you understand hypothesis as well as, the different types of hypothesis. And so to do that, first, let's look at what exactly is a hypothesis. Well, your book defines a hypothesis as a specific statement derived from a theory about a relationship between variables. Okay, that's fair enough. That's essentially what it is. You don't always really and truly have to have a theory in the sense of social control theory or the criminological or sociological theories. It's just a theory about a relationship between variables. And that theory could be, like in our example, the theory could basically be that, education majors, their grades are higher than everyone else's. Okay, it's a theory about a relationship. All right. Now, one of the things, another definition that I pulled this from the website Simply Psychology, because I want to make sure you understand there are other aspects to a hypothesis, something that goes beyond it just being something that you can, where you have a theory about a relationship between variables. A hypothesis is precise. Okay, it's clear and to the point. You don't want to put a whole bunch of stuff into the hypothesis. You might have a complex research question, but what you want to do is make sure your hypothesis or hypotheses, okay, hypothesis is singular. When I say hypotheses, that's plural. You look down here at the bottom of the screen, you'll see where I have that. So down here at the bottom where I'm highlighting, you'll see where I've put that information up there so you can see how they're written. Hypothesis is singular, hypotheses is plural. But You want to make sure that the hypothesis is precise, okay? Again, if you have a complex research question that has a lot of stuff going on with it, it may mean that you break your hypothesis up into multiple hypotheses, okay? But a hypothesis is also testable. It's a testable statement of what the researcher or researchers predict will be the outcome of the study. So in the example for the education majors, what the researcher is predicting is that education majors are going to have higher GPAs than all other students. Okay, so that's essentially falling in line with both of those definitions, the book definition as well as the definition that comes from Simply Psychology. Now, what happens is this, with regards to hypotheses, it usually involves proposing a possible relationship between two variables, the independent variable and the dependent variable. Remember, the independent variable, this is what the researcher changes. And the dependent variable, this is what the research is measuring. So in our example, let me see, in our example, the hypothesis for our example doesn't have a whole lot of information in it. Hypothesis might be grade inflation by education faculty has caused education majors to have higher GPAs. And that one, grade inflation would be the independent variable, and higher GPAs would be the dependent variable, okay? But typically, a hypothesis, usually, not always, because sometimes you have real basic hypotheses, and a lot of the examples that I'm going to use here are going to have basic hypotheses. But usually it involves proposing a possible relationship between two variables, the independent variable, this is what the researcher changes, this is what's being looked at, and the dependent variable, which is what the research is measuring. In research, there's a convention that the hypothesis is written in two forms, okay? And you see down at the bottom, that's going to be the null hypothesis and the alternative hypothesis, all right? So the null hypothesis, so here we go, the null hypothesis and the alternative hypothesis, all right? Let's see here. The alternative hypothesis is sometimes called the experimental hypothesis, or in some instances, I've heard it called the research hypothesis. So some of it depends on what field and what is going on with what's being done. So it's, again, called the experimental hypothesis when the method of investigation is an experiment, okay? So if you have someone working on an experiment or doing an experiment, that's when it's going to be, a lot of times it's called experiment. Sometimes, like for us within my field, we'll just say hypothesis. And usually when we say, what's your hypothesis, we're talking about the alternative hypothesis. I know when I write papers and when I do stuff like that, I don't write the null hypothesis. Some people do, but I don't. I usually just say, okay, my hypothesis is, and then I'll say what it is, okay? But for the purposes of what we're working on, the null hypothesis is one thing, and alternative hypothesis is something totally different. And so here they are. Here's the null hypothesis and the alternative hypothesis. The key thing about the null hypothesis, it's telling you that the difference between your variables is caused by random chance. All right. The alternative is saying basically whatever that difference is, it's real. It's not because of random chance. All right. So yeah, again, the ones that we're going to focus on here, and before we get to hypothesis testing, again, we need to understand these different types. There are other types of hypotheses that are directional and non-directional, but they essentially fall into these two general categories. And we'll actually discuss directional and non-directional hypotheses later. And that's going to be when we start looking at one and two-tailed hypothesis tests. But that's going to be, I think, like a couple of lectures later. The ones we're going to focus on here, again, is the null hypothesis and the alternative hypothesis. The null hypothesis basically is stating that there is no relationship between the two variables that are being studied. One actually does not really affect or have an impact on the other. And whatever chance or whatever changes you see, whatever differences you see between these two variables, really and truly it's simply random chance. It essentially states that the results are due to chance and are not significant in terms of supporting the idea being investigated. Now, one of the things I want you to see or want you to notice is this symbol for the null hypothesis. So you have an H and a sub zero or and that's the number zero, but it's sub zero, okay? Then for the alternative hypothesis, you have H and you have sub one, all right? So for the null, it's H subscript zero. And for the alternative experimental hypothesis, it's H subscript one. Okay, that's how we write that out, because when you are doing the hypothesis testing, you don't write out null hypothesis, alternative hypothesis. You write out these symbols that you see here on the screen. All right, you write this out and you write this out. Well, you see where I'm like making little scribble marks? Those are the things that you write out. Okay, and you'll see it when I go through and work on some of the examples. You'll see what I'm talking about. Okay, so then you have the alternative hypothesis. And this basically states that there's a relationship between the two variables being studied. In other words, one variable really and truly has an effect on the other, all right? It basically states that the results are not due to chance, all right? And that they are actually significant in terms of supporting the theory being investigated. This is sometimes called the experimental hypothesis. And this is what we're really trying to find out. But we do this by disproving the null hypothesis. I know for some people may say that's really backwards and it's really weird. That's just what we do. Okay. This is one of those times where I tell people who are looking at this type of stuff and maybe get hung up on the details, just accept it for what it is. The way that we prove the way that we try to find out whatever it is we're really trying to find out is to disprove the null hypothesis. So we want to do what we call reject the null hypothesis. That's what we want. All right. And again, so you want that null hypothesis to be untrue. And if the null hypothesis is untrue, we reject it. Okay. And if we reject the null hypothesis, then what we're really saying is that there are, in fact, statistically significant differences between the two variables. In other words, the difference that we observe, the difference that we see, it's real. All right. So like the bullet states, only one of these, it says one and only one of these explanations must be true. But the question becomes which one? And this is where hypothesis testing comes into play. That's why we do a hypothesis test. All right, so going back to our example, this is where we're going to start the process of testing the explanations, basically the null and the alternative hypothesis. Remember the education example from earlier, from a few slides ago. Now remember, the H subscript 0 is the null hypothesis, all right? So here, this is our null hypothesis, or we're trying to answer this question. What is the probability of getting the sample mean 2.70 if the null hypothesis is true and all education majors really have a mean of 3? Well, what we have to do is figure out our confidence. Remember confidence intervals? And we had the alpha 0.05, which basically said we're 95% confident that whatever our mean is falls in this interval. Well, we have a similar concept going on here. Basically, what we're saying is if the probability is, okay, let me go back. Let me say it this way. If after we do the test, the probability that our findings are due to random chance is less than 0.05, we reject the null hypothesis, okay? Again, remember, the null hypothesis means there are no statistically significant differences. So basically what's happening is you set the alpha at 0.05. And what you're saying is there's a 0.05 or 5%, okay, chance that that the differences we see are due to random chance. All right. Yeah, so just think about it that way. If you have 0.05, 0.01, 0.001, you're basically saying that, if it was 0.01, you're basically saying there's a 1% chance that the findings that we have are due to random chance. Yeah, that's when you reject the null, okay? I keep pausing because I'm trying to make sure I say this. One, I say it correctly, but also so that I say it in a way that it makes sense. Basically, you run the test, you run your hypothesis test, and if it falls within a certain region or a certain area, if it falls where it's supposed to fall, that's when you reject the null hypothesis. I will definitely say this, it makes more sense. So for anyone who's like, I don't know what she's talking about, this doesn't make sense, I don't get it, you should be able to get it after I go through and walk through some of the example problems, but also when I go through and demonstrate the problems, you'll start to see what I'm talking about. So just hang tight, hang on. And if once I get through going through all of this stuff for this entire lecture, if it still doesn't make sense to you, then that's when you can, say, hey, just get in touch with me and say, hey, Dr. Jackson, I really don't understand this. Can you help me out? All right, so what we're gonna do, this is essentially similar to what I was saying on the previous slide, but it might make a little bit more sense when you can see it out. First off, not everyone uses the 0.05 value. And remember, that's referring to alpha, okay? Remember when we did confidence intervals, we did a 0.05 alpha, and we basically were saying we're 95% confident that the number falls into this area or whatnot. Similar concept going on here. Some disciplines will use 0.10. It's rare, but it is possible. We pretty much use it for our field. So like this slide says, the 0.05 is a guideline. For our field, again, researchers typically set things at the 0.05 level. Now, think back to chapters and lessons for this next bullet. This is where a lot of you have been learning, a lot of what you've been learning will start to come together, and it'll be used to answer research questions. So, and I'm talking about the second bullet. Remember in chapter 5, when you were introduced to z-scores, also think back to chapter 7, which is where you were introduced to the alpha, okay? That's the probability of error, confidence intervals, and finding the z-score from when you know the area. All of that stuff is gonna come together here. Now there are some terms that are going to appear that you see here. They're gonna appear later on, and I'll talk about them at that point. So z-observed, z-critical, critical region, or region of rejection. I will talk more about those shortly in a few slides. But for right now, I just wanna make sure if you're looking at the third bullet and you see that, sorry about that. Okay, sorry about that, you guys. If you are looking at the very last bullet, where it says if the probability is less than 0.05, the calculated or observed z-score will be beyond plus or minus 1.96. The critical z-score. Again, there's some stuff in there some terms that are being used that you may not be familiar with. That's what I'm gonna cover those a little bit later. All right, so testing the explanations. Again, remember that if the probability is less than 0.05, the calculated or observed z-score will be beyond plus or minus 1.96. So remember, I think, okay, when we started in Chapter 5, that's when you calculated z-scores. But if I remember correctly, I think it was chapter 7. Chapter 5 gave you a few tables where you could see the most common, the most common z-scores and stuff like that. But chapter 7, that's where alpha was introduced. That's where you got the tables that said, hey, if you have alpha of 0.05, then your z-score is going to be 1.96. If your alpha is 0.10, if I remember correctly, the z-score would be 1.65. That's where that alpha comes into play. Okay, that's where that's important. All right. So now we know that since the 4.62 is beyond 1.96, we're going to reject the null hypothesis, because what you want, okay, so substituting the values into the formula, we calculate a z-score of 4.62. You don't see that wasn't done here, but when I go through and do practice problems, I may or may not do that. I may go ahead and do it just so you can see it and practice it, but we're being given that information here so that you can understand some of the aspect of the explanations. But let's go ahead and highlight that. All right, so that z-score is 4.62. And that's for the that's for our value, our 3.0, I think, if I remember correctly. Like that's what we're testing. That's what we are trying to figure out. Now, the observed z-score that we have, the 0.05, that's actually for 1.96. And so what happens is, remember how I'm always saying, draw this stuff out? This is where drawing it out. Oh, that's actually not a bad one. You guys know I have a hard time drawing these things. Oh, well, the line is a little, it's leaning, but that's okay. So this is your, remember, this is 0 because this is the normal curve. So that would be 0. Let's say 1 point, and let's say this one is positive 1.96. Well, the value that the z-score, the value that we calculated is this 4.62 right here. Well, the 4.62, because anything, let me back up. Anything that's, let me do this. All right, anything that falls over here, this is your critical region. So anything that falls over here, then that lets you know that there's a 0.05% chance. Like that's where this is where we want things to be. We want our information out here, because that's basically saying that there's not much chance that that score is going to be out here. Because remember, if we've got 4.62, okay, so because the score that we get is beyond that 1.96, it falls into this critical region here, we reject that null hypothesis. Because again, remember, we are trying to disprove the null hypothesis. We want to say that no, that score does not fall in this area right here, okay? So yeah, we do not want it to fall here. This is not where we want it to fall. We want it to fall in this area. That's the critical region, okay? All right, again, when I get to those terms, it'll make a little bit more sense, but when you see me practicing this out and doing this, it should all start to come together. In case you're sitting here and you're like, I don't get it. All this time, we've been trying to find the area in between the two numbers. Now what we want is the area beyond the numbers, okay? So just go with it. All right, so because that score fell into that area where it was beyond z-score, we are saying that that difference is in fact statistically significant. Okay, this is another way, again, another way of kind of saying what I said on the previous screen. But remember, if the probability is less than 0.05, the calculated or observed z-score, that was the calculated or observed z-score will be beyond a plus or minus 1.96, which is the critical z-score. So the observed z-score, that's the one that you calculate. The critical z-score, that's the 1.96. That's the one you want your findings to be beyond that critical region, okay? So that score, the score 4.62, it was well beyond a z-score of 1.96. So we know that since the 4.62 is beyond 1.96, we're going to reject the null hypothesis. This means that the difference is real and it's not due to random chance. Okay. So now at this point, if you are a little bit confused, take a moment, maybe rewind this lecture and kind of go back through it, draw some of the stuff out, go back and review what you've seen in the book. Take a moment to let your brain process it if you need to. But this is going to conclude part 1 of the lecture. And when I come back, or when you come back and you start looking at part 2 of the lecture, this is where you're going to actually see the different steps in the model. Because what I just did, I showed you and took you straight to what the score is at the end, but you don't know how I got that score. Okay. Initially, I said this is the z-score for a 3, and that's why I stopped and went back and changed what I said, because it's not. It's actually you get what we call the z-observed score, and there's a certain calculation that goes along with that. Okay. All right. So at this point, take your break, get ready, because you're about to get a lot of information all at the same time. All right. Until next time, take care.